All right, today we're going to continue exploring the question of how do cells control themselves? How does the DNA, which cannot leave the nucleus, tell the cell what to do? So today's topic is on translation, that is going from RNA into a protein. So we're talking about how DNA duplicates, how it transcribes itself onto RNA, and now we're looking at how does it translate itself into a protein. Again, our handy-dandy transcription translation basics. We're now in the second half, where RNA is turned into a protein. So essentially, if RNA polymerase comes along and an mRNA strand is made, this strand then goes out into a ribosome, and tRNAs, transfer RNAs, transfers amino acids, the building blocks of proteins, on together to make a protein. So that's the basics of it, the AC protein strand. So again, RNA polymerase unzips a section DNA, makes a copy of it as mRNA. mRNA moves into the cytoplasm, finds a ribosome, and the ribosome starts building up a protein strand. Let's break this down, as handy dandy as this little animation is. So again, then how do they actually do this? So once the mRNA has left the nucleus, it finds a ribosome, and ribosomes read RNA groups of 3. These are called codons. tRNA has a corresponding, so A to U, remember we're talking about mRNA, so there is no thymine, just uracil instead. So A to U, U to A, G to C. Let me circle where I was just reading that so you can see what I was talking about. So right here, these 3 letters are called the codon, the AUG or codon, and the UAC is an anticodon. It complements, in other words, what it is, and UAC happens to be for methionine, which is the start command to start synthesizing the protein. And this repeats for the dozens, hundreds, thousands of times, however large the protein needs to be, in groups of 3. And as they do this, the amino acids, these guys, are bound together as polypeptide chains, which are then built up to make proteins once they are fully translated from the mRNA into amino acids. Okay, let me just delete my circles since they don't go away when I click the screen. All right. So again, RNA, every 3 bases codes for codon. We do not need to memorize what they stand for. I will always give you that table, but you do need to know how to read it. So let's get that in a minute. So every 3 mRNA bases equals 1 codon. So you can see that up here, where GCU is 1 codon. It has code for alanine. ACG is a second codon. For threonine, GAG for glutamate, and so forth and so on. All right, this is how the ribosomes read the mRNA and know which transfer RNA to call in to build the protein. All right, so once we have that in place, every 3 bases equals 1 codon. 1 codon equals 1 amino acid. A bunch of amino acids make a protein. So how do we translate mRNA to amino acids? There are several ways to read this. I found the circle chart to be the most user-friendly, so I'll try to have handouts ready for when we do need it, or you can always Google mRNA to amino acids, and it'll give you something like this. So we start at the 5' end, that apostrophe is read as prime, and it moves out to the 3' end. So we read left to right. The left side is the 5', the right side is the 3'. This also has to do with, we talk about anti-parallel, because in DNA, one DNA strand runs 5 to 3, the other one runs 3 to 5 in the opposite direction. It can only be synthesized in one way. All right, so let's how do we read this through? So let's say that we have the codon AUG. We start in the middle, and A is first, so we start in the middle. So A, then we say, all right, then we have read to the next outer circle, U, and then G, and this lands us on methionine, which is actually one of the more important ones, as this is the stop command, the start command to start synthesizing a protein. So this is how we read mRNA into amino acids. Groups of 3 get translated into codons. And these codons correspond to definite amino acids. These amino acids are then synthesized together to make proteins. Then these proteins are folded up and either exported for use outside the cell in the environment or kept inside. Remember, proteins do all the work in the cell, all the work. Even telling when the cell went to divide in the nucleus, which genes it needs to express. So that's really the basics of it. So we translate mRNA into nucleic acid, into amino acids to make proteins. Sorry, it's been a busy morning. So that's the basics of translation. Please read your text closely, and we'll talk about this more in class. As always, send me your questions. I'll be happy to help you.